human brain organoids transplanted into rats respond to visual stimuli. On February 2, a study released in the journal Cell Stem Cell unveiled that when incorporated with rat brains, brain organoids, groups of lab-cultivated neurons, could react to visual stimulation such as glaring lights. They transplanted human brain organoids created from stem cells into rat brains. The implanted organoids were then tested with visual stimuli such as a flashing light and monitored for their response. To the researchers' astonishment, they found that the organoids similarly responded to these stimuli as a normal human brain would. Years of research have confirmed that transplanting single human and rodent neurons into rodents' brains is feasible. Plus, more recently it was proven that human brain organoids can blend with developing rodent brains. To promote successful integration, scientists grew human stem cell-derived neurons in the lab for 80 days before transplanting them into the brains of injured adult rats. Research from other studies regarding transplantations involving single cells reveals that even after 9 or 10 months post-transplanting human neurons into rodents, they still hadn't matured completely. The team is excited to see how the brain organoid progresses and is eager to test new stimuli to better understand its functionality. Follow AID for more fascinating news and updates. Thanks.